Well, thank you very much for being here. We're going to do another spotlight interview about a protocol in protocols.io called DAB-quant and an associated lab protocol article in PLOS ONE, DAB-quant, an open source digital system for quantifying immunohistochemical staining with DAB. And we have with us the 4 authors of that paper. So we have Judith Fridovich-Keil, who is a professor of human genetics at Emory University School of Medicine in Atlanta, Georgia. Galactosemia has been the focus of research in her lab for more than 31 years. Judith's training includes an AB in biochemistry from Princeton University, a PhD in biology from MIT, and postdoctoral and American Board of Medical Genetics and Genomics Fellowship training at Harvard Medical School. Judy joined the faculty at Emory in 1991. Sneh Patel is an Emory undergraduate student who has participated in a variety of research projects in Judith's lab over the past 4 years. Next month, Sneh will graduate with highest honors from Emory University with a BS degree in neuroscience and behavioral biology. This summer, Sneh will begin a year of post-bac research at NIH, after which he hopes to enter medical school and eventually become a clinician or clinical scientist. Sara Fridovich-Keil is a PhD candidate in electrical engineering and computer science at UC Berkeley, with interests in signal image processing, optimization, and foundations of machine learning. She successfully defended her dissertation yesterday. Prior to graduate school, Sara earned a BSE, also in electrical engineering, from Princeton University. Sara will graduate from UC Berkeley next month and begin postdoctoral training in electrical engineering at Stanford University this summer. And finally, Shauna Rasmussen is a research scientist in Judith's lab at Emory. Her training includes a BS in pharmacology and toxicology with a second major in biology from the University of Wisconsin-Madison, a PhD in physiology and translational science from the Medical College of Wisconsin, and postdoctoral training in Judith's lab prior to becoming a research scientist in the spring of 2021. So thank you all for being here. And I would like you to start by telling us briefly what the protocol is about, how to use it, and why it is relevant.
Sure. So I'll get us started, and then I think others will kind of pipe in. So the protocol was really devised because we had a problem in lab, which is we had slides of animal tissue which had staining demonstrating expression of a transgene. So basically, we do preclinical gene therapy for a genetic disease, galactosemia. We have a rat model, and we had these slices of liver tissue, and we could see brown staining. And if we, ground up some of the liver and did an enzyme assay, we could say how much GALT activity there was. But we know that some of the disease pathophysiology is cell autonomous. And so we needed to know not just how much activity was in, 100 milligrams of tissue, but what fraction of the cells had been converted from GALT minus to GALT plus. And so we sure didn't want to do it manually. And we did a cursory look around to see what tools already existed. And there were certainly many, but they all basically looked either complicated, more complicated than we thought we needed, or they involved a lot of user input, like the user needed to, circle stuff or do things to every image. We wanted to be able to analyze a lot of slides. And so I was moaning about this problem to my daughter, Sarah, who does image processing, and saying, well, would it be possible to come up with a program that could do this? And she was like, oh, that's easy. So that's what we did. And I will now stop talking and let maybe Sarah and Sneh talk a little bit more about how the program works. And then I'd love for Shauna to explain a little bit about, so Shauna actually prepared the slides. She did a lot, all the immunohistochemistry. And so I'd like for her to tell sort of the important parts of that.
Absolutely. Yeah.
Yeah. Maybe I can say a quick word about how it works, and then I'll pass it to Sneh to talk about how it's used, and Shauna to talk about the slide prep. So the idea is we want to quantify what fraction of the tissue that we see is stained brown in this case. And so there were sort of some technical difficulties of technical decisions of what is brown, how do you decide how brown is brown? And so I went through a couple of cycles of sort of tuning what to choose, and ultimately decided to use some negative control slides as sort of the arbiters of what is brown. So we take some slides that we know have no brown staining, and then figure out use that to set a threshold that we can then apply sort of uniformly across a whole batch of other slides. Okay, maybe I can say more later, but I'll go ahead and pass it.
Yeah, so once the software is installed on these computers, what the user's supposed to do is we put all the negative control slides in this one folder where they're processed to determine the thresholds to distinguish between empty space and tissue, and also what is considered stained versus unstained tissue pixels. And then the corresponding test slides that do contain the DAB staining and the brown staining are placed in a designated test folder, and then each scan is analyzed based on the threshold that was determined from the negative control slides, either in full or randomly selected regions, with or without exclusion of problem areas that can be defined by the user.
Okay, so then I will go ahead. So how did we get to this point? So we were performing, as Judy had mentioned earlier, some gene therapy preliminary studies to see this. And what we did is we injected our animals within the first 48 hours of life with a virus that contained GALT or our HA tag, which would be things we could use for visualizing the gene therapy and how well it got throughout the tissues that we were investigating. Subsequently, 14 to 30 days post-injection, we would perform a PBS perfusion of the animal and harvest their tissues, which were then fixated in either 4% paraformaldehyde or 10% neutral buffered formalin. We then did paraffin embedding, and we sectioned it around 5 microns thick for our slides. And we collected a couple of different tissues, but the organ we were primarily interested in was the liver, where the majority of GALT enzyme activity is typically detected. So after that, we had gone through our typical immunohistochemistry protocol, which is very similar to the general protocol that is found online, where we went ahead and we deparaffinized and rehydrated the tissue. We performed a peroxidase block and protein blocking steps. We then went ahead and put on the primary antibody, which we had for HA, which was a tag that we used to be able to visualize our GALT at the time, as a GALT antibody was not available, which a GALT antibody actually is now. So now we could also look at GALT specifically. But for this one, we just looked at our HA expression. And so we had a primary antibody that targeted HA, and then we had a secondary antibody that was peroxidase linked. And then we used the chemical imaging of DAB, which gives us the brown color, which would tell us what cells were positive, which cells were negative. And then we counterstained them with a hematoxylin, which then gives us the blue color, telling us like these cells, where they are, where the nuclei are, and whether or not there was any staining in them. After we performed that, we went ahead and we had the slides sealed, and then we sent them over to our pathology core here at Emory, which then went ahead and actually scanned the whole slide, which is a really nice advantage because you can see the whole entire tissue rather than going and getting small 10x or 20x images. We now have a full image that we can able to view at 0.1x all the way up to 80x. So we can do a lot when we get those types of scans from the pathology core. What's nice about those is they come in a file code as NDPI, which is what we use to develop the program. What's also nice is we did test additional scanners here at Emory University for other file types to see if this program would also be applicable. So we'd actually have those scans performed as well. And then we, as you can see in the protocol and in our paper, we looked at those scans to see if they would work with our protocol that Sara and Sneh developed, and we did see that those types of files would work as well. So we went ahead and we got the scans, and then we developed in conjunction with Sara and Sneh, how are we going to do this? What types of slides did we need to have in place before we went and developed this? And so we did, like as mentioned, we had the PBS injecting controls as our negative, and then our virus injector controls that had the HA gulp virus as our positive controls, so we could start to develop that system.
Thank you, Shauna.
Yeah, thanks so much. And Judy, you started the conversation by saying what were the motivations for developing this protocol and why what existed already published didn't work for you, either because it was too complex or it was too manual. But I wanted to know how the development and optimization of this protocol has impacted the research in your lab.
Well, I mean, it's a very handy tool, and we used it for studies of our preclinical gene therapy that we have already published, and we have also used it more recently. Excuse me. Before we were looking at liver. Now we're also looking at brain. So it's just sorry, it's a handy tool to have because we often need to know not just how much bulk GALT activity we have restored, but what fraction of cells have we restored it in. And down the road, we have sort of ideas about ways we may want to even extend it. I'm not sure, in general, we always have, many more ideas than we actually get to pursue. But I think that it is because it's open access, and basically people can freely modify it for their own purposes. We may modify it further for new purposes in our lab, and I hope that others will do the same.
And building on that, how can anyone use DAB-quant?
Yeah.
Okay, how about Sara and Sneh? You guys.
Yeah, I can start, and then Sneh, if you want to jump in at any point. So the code for this for DAB-quant, it's up on GitHub, and it's accessible with I think a link from the paper and from the protocol. And on that GitHub page, there's instructions for how to install all the dependencies and how to set up your slides in the right folder structure and run the code, and if you need to exclude any sort of problematic regions, how to do that. So the hope is that and all of this is just free online, so it should be hopefully easy for people to find and use. I guess I know I actually got an email earlier this week from a researcher at University of Leeds in the UK that he had found the GitHub and was using the code. So hopefully more people find it useful as well.
And another great thing that I just like to add to that is, so we did DAB staining, which is brown, but also since it's open source and it's easily modifiable, other people, if they have staining that is different colors, they can use the code to modify it for their needs. So I think that allows it to be used by a broader audience who might be doing more than just DAB staining.
Yeah, you could extend it for other colors of staining. You could also extend it for multiple colors of staining. If you wanted to stain one type of cell with one color and a different type of cell with a different color, you could get sort of richer information about the slide.
Yeah, that's one of the sort of dreams that we have now that we're looking in the brain. In the liver, we wanted to know what fraction of cells were brown. But now, we're getting greedy and saying, what fraction of neurons are brown or what fractions of astrocytes are brown. And you can do all of this with double labeling. So anyway, the possibilities are endless.
And now I have two questions somehow related, and one of them is like, you went through all this hard work of thinking about the problem and developing the protocol to tackle this problem, and now you spend more time to make it public and in a format that it will be reusable, and then the next step is to have it peer reviewed. So why did you go through that extra steps, and why did you go the extra mile?
Sure, I'll start off, and then anyone who wants to add in, please do. So science is a team sport. I mean, that's I think fundamental to the way science is and should be practiced. And so, you know, when we knew that we had come up with something that was useful for us, and we sort of by extension assumed that it might be useful for other people, we wanted to, I mean, publishing it means making it public, and we very much wanted to make it public in a way that it would be free and as easily accessible to anyone with an internet connection as possible. So, you know, the choice was pretty clear. About the peer review process, you know, we are, you know, we come from slightly different backgrounds, and we have, you know, we have this, you know, common interest in trying to develop a tool that would be useful for the purpose that we needed it. But we're not, you know, experts in all aspects of you know, immunohistochemistry analysis. And I think the whole the process of peer review, I mean, I'm a professor. I spend a lot of time peer reviewing other people's work, and I know that we help each other. I mean, nobody knows everything. And so the peer reviewers definitely brought things to our attention that we had overlooked or that we took for granted and didn't explain clearly. So I think that the final product that was eventually made public is better because of the contributions of the peer reviewers, and because we took their comments seriously and did our best to, you know, to meet them. And, you know, I think that's how science is done and should be done.
Yeah, I can give one example of an improvement that we made because of the reviewers' feedback. That we had originally developed this for the .ndpi slides that you know, my mom's lab has access to and was using, and the particular sort of computing environment that they have access to in their lab and that I had access to develop it on. But because of feedback, we tried to make it usable on both Mac and Windows and on different types of slide file extensions to try to make it so that other labs with maybe different setups could also try to use the same software.
And I'm happy to hear that the feedback that you received from the peer reviewers was collegial and helped you develop or improve, you know, the protocol in ways that it will be useful or more useful for other people. And as a service to the reader and potential user, is there anything that you tried while developing and optimizing the protocol, but didn't work and didn't make it to the final version of the protocol itself?
Yeah, I can take a stab at this, and feel free to jump in, anyone else, if you want. So we did, one thing we tried doing, so Shauna makes beautiful slides. And it turns out when we started trying to work with different slide formats, it turns out different, like, there are some aspects of the code that were sort of developed with Shauna's slides. And for example, one of the other slides we tried working with, someone had written in Sharpie on the slide, and the code got confused.
Not us. It was the scanner. Someone in the scanner put a mark on it.
We had tried coming up with a way to, like, manually crop the slide, or in the code crop the slide to exclude sort of unexpected things like that. And it ended up being a bit trickier than we bargained for. So we did not end up including that, but I guess you could always manually crop it before feeding it in.
Another thing I would just like to add, I don't think this, like, it did make the cut for the software, but Shauna mentioned how we decided to include the different file formats because we had access to the .ndpi files, But for the other ones, because of just not having good quality slides, I think we weren't able to really test our software for those file formats, even though with the code and the Sarah could probably talk more about it, but like the fact that adding a plugin that was able to open those other file formats, theoretically, we know that we'll be able to use this for different formats, but we weren't able to really test it because of just the lack of access to those different file formats. But I think that's another thing that we tried and was definitely an improvement based on the reviewers' comments.
And are there any steps of the protocol or parts of the protocol that are particularly tricky and where users have to be extremely careful when running it?
Yeah, I can start off with this and then other people could jump in. So I think the most tricky step, in my opinion, is to set the error tolerance. And what we mean by that is the user can put in an error tolerance percentage. For example, the default is 0.05%, which is then used to determine the threshold, the normalized red minus blue threshold that deems a pixel to be stained or unstained. So if you look at the distribution of the normalized red minus blue pixels scores for like a negative control slide, like a 0.05% error tolerance means that no more than 0.05% of those pixels from the negative control slides are going to be deemed to be stained. So there was a bit of subjectivity in choosing that error tolerance for our purposes. So our software produces these pseudo-colored images that converts the brown into pink and empty space into green to, I guess, better visualize these images. So what we were doing is looking at these different regions and then seeing, is this error tolerance capturing what we want? And so we had to use some of the subjective and eyeball testing to determine that error tolerance that worked for our set. But I think for other users, they're going to need to do some experimentation to determine what tolerance works for them. But also, I guess on a positive note, like looking at these different error tolerances, if your research question is looking into the different gradations of staining, because there could be light staining versus really intense staining, it could give you more information on that. And also the distribution of the normalized red minus blue scores could also give you insight into that. So I would say that's the most tricky part that the users would need to be a little careful about.
Yeah. One other sort of thing to add on to that is that it's important that the negative control slides and the positive, test slides come from the same batch. So we'll get sort of consistent, whatever threshold you pick will be consistent across the whole batch. But as you vary that threshold, the sort of absolute numbers might change, but you'll get generally a consistent, at least, ranking of which slides are most stained. Yeah, across the batch.
And I'd like to add that when we first started sort of grappling with the idea of where should we set the threshold, how brown is brown, and when I first started doing staining back, in the mid-Jurassic period, when I was a graduate student, we used the eyeball test. It was like, well, if it looked stained to you, then it was stained, and if it didn't look stained to you, it wasn't stained. And then when we started looking, some of our samples for biologically valid reasons have quite a range of staining, and it made me realize that, maybe what the human eye or what somebody thinks looks brown, maybe that's not like a magic, threshold that we should be trying to make the computer have the same limitations as a human eyeball. Maybe the computer can be better than the human eyeball. And then it really, it made me realize that, there are very few absolutes, and that you really have to decide. So we wanted a mathematically defensible threshold, not just a, well, it looks brown to me, threshold. But I think we struggled with that a lot in terms of how should we address this. And, it is certainly user, the user can set the threshold wherever they want, but that means that the user really needs to be mindful about how they're doing it, because if you set that threshold high, you can say, I got 50% of the cells are brown, when in fact, that's probably not true. So I think you need to be careful about it. And as a reader, you also need to always be, check those materials and methods and see, and that's not just for our system, that would be for any system, of analyzing stained tissue. Everybody's going to have to deal with threshold one way or another. And so how did they do it is an important question.
And you have already touched slightly on like all the possibilities, and all the idea that one could have about this method, and there are usually more than what you can actually do. But where do you see, or where's the potential of this method to develop for other applications, or to further optimization, if you can have a wish list?
I'll give my wish list, and then anyone else can pipe in. So, as we mentioned before, I think the idea of having double staining and having the system be able to, so we might be doing, let's say, I don't know, I'm thinking in terms of immunofluorescence. The reason we did immunohistochemistry is that the slide scanner that we have access to can't handle fluorescence, but I'm sure that there are others that can. So if we could have the system look at different channels and look at maybe the red channel and the green channel, and we stained for neurons with the red channel and for transgene with the green channel, and so that we could basically have we could expand from that and have the system tell us not only what fraction of pixels are brown, but what fraction of pixels that are green are also brown. I think that would be very powerful. So that's one thing on the wish list. And another thing is we would love to be able to, now that we're looking at brain, it's a lot more compartmentalized than the slices of liver we were looking at. And we would love to be able to say how much of the brown is in the cerebellum and how much of the brown is in the cortex. And so, there are different ways that we could, either, have the user trace around the cerebellum and say how much brown is in there versus, so I think there are a lot of ways this could be done. We haven't really thought about it deeply enough to do any of it, but I think there are a lot of options, and hopefully other people will have yet other ideas that could make this further useful. Anything else? Okay.
I was gonna say, I don't comment and second what Judy said, being able to do the different channels and colors would be great. I think it'd also be very good to be able to see, if there's a tissue type that doesn't work in this system, which I would be surprised by. And the ones that we were performing with were, paraffin embedded and stuff like that. There's a potential to use maybe frozen sections or something like that and get an image from frozen sections. Since a lot of neural stuff with the brain are done with frozen sections versus paraffin embedded, that would be a great addition if we could somehow get frozen sections imaged and be able to use the software.
Or even higher resolution images. Yes. So instead of looking at cells, we were looking within cells to say how much of the staining is in the nucleus, how much of the staining is in the Golgi. So I think there are a lot of possible applications. And at this point, this was kind of a side project for us, so we probably won't actually do them until our science drives us to need that functionality. But I hope that other people will be able to take what we've shared and tailor it for their specific needs and then share that back so that the scientific community will have an increasing tool chest of things to use for science.
I can maybe add one more wish list item that I'm not totally sure how well this will work, but it would be great to have, would be if we had a sort of fair way to compare across different batches. So the thing I would sort of naively want to do is, well, take control slides from this batch and control slides from that batch and pick the same error tolerance threshold for both of them. And that'll potentially produce different actual brown thresholds for the two batches. It'd be great to sort of verify in some way that these are actually making the numbers comparable across the two batches. It seems sort of possible, but we have not actually done that verification.
Yeah, that's good. I'd like to add one thing. One of the functionalities that Sara built into the program, because I was really worried that slides would have bad imperfections in them, places where the tissue was folded or wrinkled, that stained aberrantly because of that imperfection. And I didn't want that to, confound our data. And so we wanted to be able to exclude those bad regions of a slide. And so Sara went to a lot of trouble to build that functionality in, and Sneh went to a lot of trouble, testing it. And Shauna makes such great slides that there were very few wrinkles or problems, so that after Sneh went to great pains to eliminate every box that had fallen anywhere near a tiny wrinkle, it hardly changed the answer at all. So I think I'm still glad that we have that functionality in the system, because some other tissues that someone else is using might be more vulnerable to these, imperfections, or they may not. The person who makes their slides may not be as talented as Shauna. I don't know, but so that was sort of something I really thought that was going to be like, oh wow, this is going to be great functionality. It's going to really clean up our data, and then it was like, didn't really matter. So.
But that, on the plus side, it was sort of robust to that.
Yeah, and that highlights the importance of having a good starting material.
Oh God, yes.
Biological material has to be high quality.
Yes, that is a key aspect of this protocol. What you want to put into it needs to be of good quality. And anyone that does histology knows that it can be tricky. Certain tissues are easier than others. Some like to fall off a little bit during the processing stages. So it's really important to try and have the best quality of slides being put into the system. Yes.
I can't tell enough how much I have enjoyed this conversation. And I think that we have talked about, the protocol itself and about making it public, different angles in this conversation. And all of you are in different stages of your scientific career. So I would like to know what has been the significance of getting this protocol done, publicly shared, and published as a formal peer-reviewed article for each of you.
Who wants to start? Sneh, why don't you start us off?
I can start us off. Yeah. So I think I'm in the earliest stage of my scientific career here. So I've been very grateful for this opportunity as like an undergrad to be a part of a project that is very different from what I anticipated I would get into when I started working in Judy's lab. So I think I've been very grateful to expand my biological knowledge in addition to this technical knowledge about histology and immunohistochemistry. And I think as I graduate and move to the next step, whether it's going to NIH or eventually going to medical school, like I look forward to building on this foundation and using this knowledge of histology and IHC and IF and staining and all this as a clinician scientist. So I've been very grateful, and it's had a significant impact on my journey as a scientist. Yeah.
I guess we can go in reverse seniority order. So most of my research is quite different from this. I work in image processing and electrical engineering and computer science, but most of the types of image processing that I'm interested in have scientific or biological or medical applications. And so this was sort of a rare opportunity for me to be able to actually see how image processing can actually be used for advancing, medical science. So I hope that as my career goes forward, I'd like to develop more collaborations where I can actually try to have impact with image processing in these applications.
So being someone that's in like the early middle stages of their career, having a tool like this is really important for someone like me, who is doing a lot of different aspects of our research, to have a tool that I could essentially upload, get organized, and then walk away from for a day and do other things is really important when you have a lot of other tasks to be done in the lab. This would have been a tool that I, as a grad student, would have loved to have had for my dissertation work, where I was, looking at transplants of GFP cells in the liver of rats and stuff like that. So I would have loved to have been able to just upload all my slides and had all this data computed to me, instead of me having someone else go in and draw the boxes, where then we have the introduction of potential bias. This program is nice because it removes that potential bias of selecting certain areas of a slide and stuff like that. And I see this tool being utilized quite a bit in our lab as we continue our research into gene therapy and other potential therapies for our patients in our rat model.
Thanks, Shauna. So I guess I'm the old one here. So I'll say for me, I have, I've been a PI for more than 31 years, but it never gets old. I love learning new things, and I get a great deal of sort of pleasure and excitement out of seeing, okay, we're trying to accomplish a certain goal. We are using a certain approach. We hit a brick wall. Okay, what are we going to do? Look around to see, is there already a tool that will get us over, under, or around this brick wall? And if there is, use it. But if there isn't, or we don't know how to find it, sometimes you can make your own ladder. And I think it was really fun for me to get to work with this team of wonderful people, to basically make a tool, use it for the purpose that we wanted, and then say, hey, I bet other people would like to use this tool also, so let's share it. And so it's part of why I love doing science, and this project was really a fun one.
Thank you very much. And before we wrap up, is there anything that you would like to mention that we haven't touched?
You've done a great job. Thank you.
I looked at my list. I think we've covered, all the very important aspects in, the steps that we've taken. So I don't really have anything else to add.
Yeah, so thank you for all you do to help make science public and accessible, and also I think to show sort of the human side that science is done by people, and we are imperfect, but we're doing our best.
So then I will finalize the recording now.
Okay.